Again, I would like to welcome everyone on this call to this day of lung science. This is a webinar organized by the McCary Lung Institute, usually to give us more knowledge about what is being done in lung health. I would like to welcome everyone on the call. My name is Treasure Iringira. I'm your moderator today. I'd like to also welcome Professor Chirenga, and all of the people that have spare time to join. So today we shall be hearing from our esteemed Dr. Immaculate Nancha. But before I let her take over, I see we're already late. I want to talk about MLI at 10 years. Could you help me upload the slides on MLI at 10? If not, I can just briefly tell you about that, but let's wait and see. So McCary Lung Institute is celebrating 10 years this November. It will be marking 10 years since its establishment, which goes back to 2015. Among the founders of this Lung Institute is our very own Professor Bruce Chirenga, who is also our principal. And we would like to recognize that achievement. Are the slides ready yet? So because of time, let me introduce our speaker today, and then we can get on and catch up with the program. So our speaker today is Dr. Immaculate Nancha. She is a doctor who also holds a PhD. She is currently the on-site director of the Center for AIDS Research at the Joint Clinical Research Center. She is a molecular biologist with major research interest in genomics. So currently she is undertaking research in HIV drug resistance in both adults and pediatrics. She is also undertaking research in the area of HIV cure in various populations, including pregnant women, newly diagnosed individuals, adolescents, and young adults. She is also currently doing research in cancer genomics, identifying biomarkers for targeted and precision oncology. And maybe lastly, together with a team of other experts from the laboratory field, she is utilizing cutting-edge techniques to set up a state-of-the-art precision lab in line with precision or personalized patient care. And this is in areas of oncology and in vitro fertilization and also non-communicable diseases. So with that brief introduction, I would like to welcome Dr. Immaculate Nancha to currently tell us about the topic for today. Dr. Immaculate, the floor is yours. You are most welcome.
Yes, thank you very much. I hope I'm audible. And thanks for that introduction. Good afternoon, everyone. Already cancer of B in the era of precision. So I would like to point out that everything today is going to be molecular. So I'll try as much as possible to make it digestible, easy for you to understand as much as I can. So this is just a summary of what I'll be looking at. Molecular mechanisms of oncogenesis, and then what are the factors triggering lung cancer, molecular pathogenesis of lung cancer, the biomarkers involved, and how are they detected, what are the innovative approaches for personalized care? That is very important because that's where the field is going. And then, of course, when someone is on treatment, there is bound to be resistance. So what are the mechanisms of resistance to cancer drugs? So I'll start off with something easy, and this is the global prevalence of cancers. And as you can see, lung cancer accounts for 11%. It's the most common, second most common cancer after breast cancer. So that is of clinical importance for us. And as we all know, maybe not know, but there are risk factors that predispose people to lung cancer. And key of them is smoking, that is in the smoking world. But in our world, we also have smoke that comes from maybe cooking, move from wood and from everything else that exposes us to smoke. Then we have exposures from metals and radiation, and then occupational. Then we also have viruses. We've recently seen that one of the sub-studies from this study have shown that HPV is one of the viruses, and it accounts for about 20% among the lung cancer patients we have so far looked at. We have also seen that the most common cancer here is the non-small cell cancer, and it's more common in females than in males. So what is the mechanism of oncogenesis? This is in general. We know that oncogenesis is multi-step, and it involves turning healthy cells into cancerous cells, and each step adds to these cells the ability to become more and more cancerous. So also we know that in the cell, there are genes that make sure that the cell is in order, that every function goes on well. In the genes, these are genes that are normal and function normally. And then we have also genome maintenance genes. In a normal situation, everything is normal. But in cancer, these genes get alterations or mutations. And once they get these, this triggers cancer development. Also in normal beings or in normal cells, there is what they call cell death. There is cell aging, senescence, and also growth arrest. These are normal procedures that keep the human being or the cells in check. But when there is a disarray, these functions like cell death and senescence and growth arrest all come to a standstill, especially in cancer situations. So this is just a cartoon showing you a normal cell. Like I already mentioned, the genes in there are doing what they are supposed to do, and you have a normal cell. Everything is under control. But when you have a cancerous cell, there are mutations within those genes, and then this causes uncontrolled cell division, and then you have all these, cancerous cells coming up. And this comes up, there are factors behind it. We mentioned these in the second slide where we have all those predisposition factors. So here we have a few of them that I'm going to outline. We talked about also the carcinogenic exposures. And when you have these exposures, the chemicals lead to DNA damage. DNA is the building block of all genes. So when there is DNA damage, then there could be, maybe there are deletions within the DNA, then there is cross-linking, maybe there is one strand is removed. So all this causes like a disarray within the DNA, and it triggers the cancerous process to start, or oncogenesis. We talked about viruses, and in our case, HPV is one of the culprits. So when there are these viruses within the lung environment, you find that the immune cells will definitely come in, try to destroy the virus. And then when they do so, they release certain chemicals that are mutagenic, like the oxygen-free radicals. And these are key players in the oncogenesis. Also, we have the tumor microenvironment. And this is mainly, tumor microenvironment is comprised of tumor cells. They are the stromal cells, and also they are immune cells. And this all comprise that. But then also there is what they call the microbiome. Microbiome are the microbes within the tumor microenvironment. They play a very important role in also triggering the cancer process. And how do they do that? we see that recently there's been great interest in the microbiome, the microbial environment in every organ. So in this case, we are talking about the lung, and it has been shown that it plays a critical role in the human health, both via immunity, metabolism, and inflammation. So the microbiome is very important. And now when we look at the microbiome per se, we have the oral microbiome, we have the lung microbiome, we have the gut microbiome, and all these are in constant, they mix, there is connection, they are dynamic. So microbes move from the gut to the lungs, they move from the oral cavity to the lungs, they move from the gut to the oral cavity. So with this free flow of microbes, then there is free flow of those microbes that will cause cancer. And studies have shown that some microbes within the gut have been attributed to lung pathologies, including cancers. So we see that with the microbial dysbiosis is whereby normally we have the microbiome, it is in sync with everything else, and the microbes are okay with everything else going on in the system. But when there is dysbiosis, the microbes are out of order, and then they trigger inflammation, they trigger DNA damage, immune suppression, and this ultimately cause tumor growth. That is why the microbiome is very important, because it ultimately leads to tumor growth. So microbiome composition affects how patients also respond to therapy and immunotherapy. So there are certain bacteria which may enhance response to certain drugs, while others may inhibit that response. So we see that still the microbiome is playing an important role. And when we are looking at the microbiome, especially in lung cancer patients, we are looking at various microbiomes. So we look at the tissue microbiome that is in the lung. This directly affects whatever happens in the lung. It will influence the immune response. It will influence tumor growth. It will influence response to therapy and immunotherapy. Then we also have the microbiome within the pleural effusion. And this also, it's an indicator of what is going on in the tumor microenvironment. We already talked about what that involves. It has the stromal cells, it has the tumor cells, it has the immune cells. So the pleural microbiome is also very important. Then we have also the bronchoalveolar microbiome that is in the air spaces. Then we have the gut microbiome. I talked about this, and it is known as the gut-lung axis. So these microbiome, when they move back and forth, they've been implicated in all these pathologies in the lung cancer. So this just explains what I've been talking about. You have a homeostatic lung where the microbiome is in sync with everything else, and there is peace. Then, if there is dysbiosis, there is an imbalance in the microbiome, there will be production of toxins, there will be production of cytokines. These will cause inflammation, they'll cause DNA damage, and then you'll end up with cancer. So that is how we have these two working together, either for good or for bad. For bad is when you have the cancer. So after we've seen that pathology, then we have to what actually happens when what is the pathology or within the lung cancer? What happens there? So what happens exactly is there is accumulation of mutations, genetic mutations. Then there is also uncontrolled proliferation. The cells are uncontrollably proliferating. There is also escape from apoptosis. Apoptosis is it is a cell check mechanism whereby maybe the cells, if the cells are not, they are maybe infected or they are pathological, they are targeted for destruction. But in the case of cancer, you find that these cells are escaping, being destroyed, and so they keep around and they keep on proliferating. Then there's also overexpression of angiogenesis. So the blood vessels within those tumor areas are like very vascularized, and you have these supplying more and more cells and growing and causing more and more tumor to become bigger. There is also unregulated replication and also the ability to invade adjacent cells and tissues, and also for distant metastasis. That is how cancers develop, spread. So now the key pathogenesis, or how it all starts, is in this cartoon. It may look a little complicated, but it starts off with the EGFR receptor right here in purple. This is, it is called a transmembrane receptor. It is across the membrane. As you can see, there is this membrane and it's across. So this receptor is a natural receptor. And in normal instances, it will do what it's supposed to do. When a ligand binds to it, the EGFR, then it will, the EGFR ligand, when it binds, it will just go ahead and go into signal transduction and stimulate different genes, and then you have everything normal in the cell. So it can take the RAS route, that is the green, and you have all these genes being stimulated and you have everything going normally. Then it also can take the other route where you have light blue, but it's also normal. But when there is a mutation, for example, in the EGFR, the receptor itself, or along any of these genes, the RAS, the MEK, whatever, the MAP kinase, or any of the genes along the pathway, if there are mutations, then there's bound to be a disarray. So there will be uncontrolled of everything. There will be uncontrolled proliferation. There will be invasion. There will be angiogenesis that I talked about, many blood vessels supplying the neighboring and causing the tumor to grow. There is metastasis. There is also apoptosis, evasion. So these cells will not be able to go through the apoptotic pathway to be destroyed because apoptosis is cut off. So this is how now what happens, and that is how the mass becomes bigger and bigger because everything is uncontrolled and it cannot be killed, programmed to die. So we already talked about here, these genes, all these genes that are in this cascade are the genes that contribute to the markers that we see in lung cancer. On the left, I think, the genes, as you can see, there is the ALK, there's EGFR, there is KRAS, all those genes are there, and they have their roles. And these are the markers that we look for when we are assaying for any lung cancer patients to see whether there is lung cancer or not, or if there will be response to treatment. So when you have a biomarker, which is a gene, and there are mutations within that gene, then the role of that gene is also indicated. Then there's also what you call targeted therapy, where you have, for each of these markers, there are drugs. I'll talk about this later. So we have seen those genetic biomarkers, but also there are other biomarkers, like the protein expression biomarkers. We have the PD-L1. This one is the anti-BTL drugs that have been developed for immunotherapy. We have circulating biomarkers. This is now the new technology where you have circulating tumor DNA, and this is found in blood, and it is used for non-invasive testing, that is diagnosis and monitoring of treatment. These I'm going to talk about more in detail. Still under circulating biomarkers, we have the exosomes, and these also are key. We'll talk about them. And then we have also what is called a tumor mutational burden. This is also a key marker that we look for when we are, when you really want to know exactly what the patient has, so that you can help them to have like more precise care. So I'm going to just talk about the tumor mutation burden, because it's very important, but I'll also talk about the others. So tumor mutation burden is the number of mutations, somatic mutations, per 100 base pairs of DNA in a tumor. So they measure it with next generation sequencing. It is a sequencing platform that does this, and you see how much of the burden, the mutations are there in this tumor. But then why is it important? Because it is also a predictor of treatment. When you have a high tumor burden, these mutations, they have what they call abnormal proteins. They are the neoantigens, and these are recognized by immune system. So that makes this very important, because now immunotherapy can be used, and they call them the immune checkpoint inhibitors, can be used for immunotherapy, because now this patient will be responsive. But also if the patient has high tumor burden, it means that a high mutation burden, it means that there are so many mutations, and you find that it's almost impossible to have a precision regimen for them, because it is like everything is knocked off. So, and actually there are drugs that have been developed. There is a drug that is approved by the FDA based on the tumor mutation burden. And this is used for all solid cancers, regardless of that type. So that's why tumor mutation burden is very important as a marker. So, okay, so definitely we need to go into, once you have a cancer, you know how it is evolving within the patient. We need to see how to detect it. Early detection is very important, but unfortunately in our settings, most of these patients report very late. And so most of this may not be applicable, but it's good to know what is there so that in case someone comes early, then you know what to do. So there's what they call exosomes. I'm going to talk about them. This is emerging technology that is used for early diagnosis and also for prognosis. And what are exosomes? These are small vesicles. They are produced by the cells, and then they bud off the cell, and then they are called molecular messengers. They carry they carry cargo. And in the cargo they carry, there is nucleic acids, that is DNA, there is RNA, there are lipids, there are proteins, and there is everything. So they carry these from one cell that has released them to another cell that is receiving them. So this is just a cartoon to show what an exosome is. You can see that the zoomed out thing, this one, the zoomed out one, this is coming from the cell of origin. So the cell of origin has released exosomes, and one of the exosomes is zoomed out. And what is contained in there is DNA, there is RNA, there is lipids, there are metabolites. So it has everything in there. And it's carrying it from one cell, the cell of origin, to the recipient cell. So this is a very, very key mechanism, because these exosomes, they mediate intercellular communication. They're coming from one cell into another, and they are releasing their components from one cell into another cell. And this is also important for cancer spread. You see that as it's releasing, if it's coming from a cancer cell, it will release whatever it has got from this cell to another. And that is where it causes metastases. So exosomes are very important in that area, but also as transport molecules, they are important because I said that they carry all these things. And another important thing that we'll see is that they are of low immunogenicity. They do not attract an immune response. And also another thing is that they cross barriers like the blood-brain barrier. So even for spread, they can easily spread into, they carry these contents into the brain. So you'll find that there are metastases in the brain of a certain cancer, maybe not for lung cancer, but I'm just bringing this in context because this is new technology that is coming up. So why do they really matter? I already talked about their communication from one cell to another. Disease progression, I already mentioned that, because they spread, say if it's the cancer cell to another, they'll take the content of the tumor DNA to another cell. And then also they are good for diagnostic purposes because they have the diagnostic specific markers. So if you're looking for, say, a patient who has lung cancer and you're looking for mutations, you can use these exosomes to screen for those markers that are really present. And the advantage of this is that these are released into blood. So they are released, the cells release them in, cell to cell, but within blood. So the media they use is blood. So when you screen blood, you ultimately find the exosomes there. And that's where we get also the concept of circulating tumor DNA, because the exosomes are many in the blood or in other blood and other body fluids. So you get them, and then you can use them for screening purposes or diagnostics. And also they are important for therapeutics. Scientists right now are into the process of using them as delivery tools, because we said that they transport cargo. They can as well transport drugs to the target cells. So that is quite impressive. So still on the exosomes, because they resonate, in that we talked about the tumor microenvironment, and it has the tumor cells, it has the stromal cells, and it has the immune cells. So these exosomes carry stuff from all the tumor microenvironment. And so within the tumor microenvironment, you will find that you have exosomes from immune cells. And because they are immune cells, they have immune components in them. And you have exosomes from tumor cells. And because they are tumor cells, you will find their drug-resistant mutations there. You'll find that there is excessive proliferation. You find that there is stemness, resilience. There are those cells that the components that make the tumor resilient and non-responsive to treatment. So you have those tumor cell exosomes, and then you have also exosomes from the stroma, and these have fibroblasts. So all this is happening within the tumor microenvironment, and this all affects how the cancer progresses. So even in the context of lung cancer, this happens within the tumor microenvironment. And you have an interplay of all this working together to cause the cancer to grow and progress. The immune response may come in and try to fight, but sometimes it becomes overwhelmed. So now, how do we detect these biomarkers? We talked about the biomarkers that are genetic. We've talked about the biomarkers that are proteins, and then those are in circulating tumor DNA. We have talked about biomarkers in exosomes. And so how do we detect them? So we use the biomarker, okay, that is, we detect them, and so that to provide critical information on cancer response. We can also use that biomarker detection can be used for cancer diagnosis, response to therapy. We can also use it for like a screening tool. For example, you can use EGFR as a screening tool for predisposition. Like if someone is highly predisposed, maybe there is a genetic predisposition, you can use it as a marker and screen it, and then just know that, okay, this patient may not manifest cancers yet, but there is a predisposition, and because of this mutation, they are most likely to have cancer. So that's why we do the biomarker detection for treatment, response to therapy as your monitoring response, and also for screening purposes in those who are genetically predisposed. So how do we do this? One, and the most, right now, that because we're in the era of precision medicine or personalized medicine, the mainstay of personalized medicine is next generation sequencing, where you have, we talked about those genes and those markers and all those genes involved in the cascade into a panel. And this panel may have about 50 to 60 genes. And because we don't know what each patient has, everything is put in there. And then we do targeted sequencing. We target those genes and then sequence them and then see which of these genes out of the 50 have mutations, whatever mutations. For example, in lung cancer, what mutations are there? What genes are affected? And if it's a gene, what mutations are within those genes? So next generation will be able to tell you that. And also, it will be able to give you options for treatment. So for example, if the patient has EGFR, maybe a deletion here and there, then it will give you the most appropriate treatment you can give that patient. And because each patient has different mutations, this is the best way to give precision care, because everyone will have different and everyone will have their own medicine as opposed to a one-size-fits-all. So then there is also IHC, where you have the PDL, the programmed death ligand, and this is also for immunotherapy. We have FISH. As a lab, we don't do FISH, but as a lab, we do NGS. That's what we are doing, and we've done it for quite some time now. There is also quantitative PCR, where you quantify particular biomarkers, for example, EGFR, KRAS, or MET, or whatever it is. So whatever marker you want to, you can quantify it. And you can use this, one for genetic predisposition as a screening tool, but also you can use it as you monitor patients responding to treatment. So you can use quantitative PCR, but there's also digital PCR. And digital PCR has the ability of detecting low-level circulating tumor DNA as found in exosomes. These exosomes are tiny, and so the DNA in there is not so much. So you need a sensitive assay that will really pick out that and use it as a marker, and so point out that this and this is present. So all these assays, we have them in the lab, and we have been doing them for other approaches, but they can be used also in cancer monitoring and also diagnosis, as well as screening. And liquid biopsy is also, yeah, it's a method, but it's a less invasive method where you get the blood sample, and because we talked about exosome releasing or circulating and moving from one cell to a distant cell or wherever, so you have them in the in the blood, and so when you sample the blood, then you can easily get those out and use the circulating tumor DNA to assay for whatever markers you're interested in. So I'm just going to show some results. This is a study that we are winding up this month. It was a 5-year study, and it's closing down right now. And we had 2 cohorts, one in Uganda at the Lung Institute and Cancer Institute, and then the other one in Tanzania, and wanted to look at the prevalence of mutations among lung cancer patients. As you can see, EGFR is the biggest player, accounting for close to 80%. And you have to remember that our population is mainly non-smoking, like tobacco or cigarette smoking. But it's also smoke from other sources, but it is a non-smoking, cigarette smoking population. So this was the most predominant, and it was comparable between Uganda and Tanzania. And also the other mutations are there, the ER, BB2, and all the other mutations. So when we compared what we found in our East African populations and compared it to Asia and Europe, we found that EGFR, like the prevalences, were almost comparable to those in India. We had 77%, and India has like 45%. That's what the data shows. However, in North America and Europe, because it's a smoking population, it is 15%. So then we looked at the smoking population, at the bottom is KRAS. This is the most predominant in the smoking population, and it's about 30%. So we compared the populations, and we saw that our population is closely related to the Asian population, and most of them are non-smoking. Then also this is part of the results. So okay, we looked at, like, we know that these are genes, and within genes are mutations. So when you look at the EGFR gene, we just teased it down by mutations that are found there. And the most predominant one, as you can see, the tallest bar, was the deletion at exon 19 in EGFR. But then there were also other mutations there. And then we also had mutations in the KRAS, that is the one for the smoking population. Although in our population it's not so high, but it's also there. Then we had other genes. So we see that there is a multitude of mutations. And so when you look at this, definitely you cannot say that I'll give EGFR inhibitors. When you do that, then you're missing out on the KRAS mutations, you're missing out on the phosphorinositidil kinase, you're missing out on the MAP kinases and all these other things. So that is why it is very important also to do genotyping, so that you know exactly what the patient has, and then give treatment based on what you have found. And genotyping is the key for precision medicine, or precision oncology in this case. So I already shared this slide, but I wanted just to point out that for all these mutations, they have what they call actionable. Okay, they are actionable mutations, meaning that there is an action for them or a drug for them. So when you look on the farthest side, on the right, you will see that all these biomarkers with their mutations, there is treatment for them. So if you do precision diagnosis, then you can get precision treatment for these patients. And this is very important. So now that we've seen the mutations and the treatment options, definitely there's bound to be drug resistance that happens, either intrinsic or acquired as the patient starts on treatment, but it happens. And why does it happen? One, it's because cancer cells multiply or they mutate so fast. So when they mutate, they escape. And the treatment that has been designed can no longer be effective. So next, there are those glycoproteins that pump drugs out of cancer cells. So you have a patient, you put them on treatment, and then these cancer cells develop proteins that will just pump the drugs out. And once the drug is out, then you have insufficient drug, and then you will not have response. So then also there is what they call tumor heterogeneity, whereby there are various populations within the cancer cell, the tumor cell, the tumor. So these cells are various. So you may have these that are responding to treatment, but the others that are just naturally not responding because they are heterogeneous. They are not conforming to the structure of what the cancer drug is targeting. Then there are also activation of alternative pathways. So once you give that drug, then the cancer cells will bypass that pathway, because we saw these are pathways, signaling pathways. So if you give a drug and it blocks that pathway, the cancer the cells will now activate alternative pathways so that they can still continue to multiply. Then there is what they call enhanced DNA repair. So when you give these drugs, and then there is maybe DNA damage and it's being fragmented and all that stuff, the cancer cells will put a mechanism in place to fix the damaged DNA so that they can still continue to multiply. There is what they call epigenetic changes. Epigenetic means there are those modifications like methylation, where you'll have various methyl groups on the genome. So once you have these added groups, so the genes that are supposed to respond to treatment are silenced. And this is a trick that the cancer cells will do also. So response to therapy, and then they continue to multiply. Then the tumor microenvironment, we talked about that, where there is the stroma cell, the tumor cells, and all that things. So you find that these fibroblasts within the stroma may protect the cells. They protect the cancer cells. They just put a hedge around them so that the drugs do not get to them. Or they may secrete factors that will make that will promote resistance. Okay, so I'm just showing this. We talked about methylation and how, there can be modification of the genome. And once it is modified, then there is silencing of those genes that are supposed to cause sensitivity to the drugs. So this is one of the results from our study as well, where we compared methylation in HIV-positive lung patients and HIV-negative lung cancers. And we found that there was more methylation, or there was more modification of the genome in HIV-positive patients than in the negative patients with lung cancer. So this is also an important escape mechanism. So I already talked about the types of resistance. It is intrinsic. It may be intrinsic, like where you have heterogeneity, some of the cells are responding and others are not responding. Then it can be also acquired, where the treatment itself now causes the cells to just find an escape mechanism. So if there is resistance, how do we overcome it? One of the mainstays of cancer treatment is combination therapy. You never find a patient getting a single drug. And the reason that is done is because you want to kind of like surround the cancer so that whatever mechanism they are using, you're taking care of it. And that is one way. Another way of also overcoming resistance is to use targeted delivery, where you use nanoparticles. Nanoparticles are small particles, and they can carry drugs at certain concentrations, and then they concentrate it in that site of the tumor. And if it's concentrated there, then the chances of killing the tumor cells is very high. And now the mainstay is the precision medicine. That's why we do genotyping. So once you do genotyping, you are precise. You know exactly what is going on, then you target it. And even after you've done the precision medicine and you've given the patient the best medicine, there is need to monitor response to therapy, because some of these arise during the course of treatment. So there is need to take off serial samples and then monitor and assay them for biomarkers, especially if you did a baseline where you have done the genotyping and you know that there are genes, EGFR, there is KRAS, there is this and the other, then you can pick one of those as a marker that you're going to use to monitor response to therapy. Then also there is immunotherapy, where you boost the immune system so that it can recognize and destroy resistant cells. So now we go into there's so much research going on right now in cancer, and cancer cuts across. So there is so many approaches right now for personalized care or precision care. And one of them is being less invasive. So we know that for the mainstay of diagnosis of cancer is by biopsy, but we have those biopsies that are out of reach. For example, lung cancer, you have to use CT-guided biopsy, and then you get the biopsy. So once you get the biopsy, then you do the diagnosis, whichever diagnosis you're going to do. But after you've done the diagnosis, you may need, okay, you started the patient on treatment. How do you monitor? If it is like lung cancer, how would you go back again and again and you do these biopsies from the patient? It is not possible. So studies have found out that you can do less invasive methods, and that's where circulating tumor DNA comes in. And that's where the exosomes come in. So you have the exosomes circulating the blood, and then once you take off that blood, it is representative because these tumor DNAs are being taken all over the place. So once you get them, then you can use them, and this is less invasive, and it is more effective because you'll be able to monitor your patient while they're on treatment. And also to come with this, another innovative way is also, because I mentioned that exosomes carry small DNA quantities. So it is hard to accurately measure them with, especially like qPCR, quantitative PCR, but there is now a newer technology that is digital PCR that quantifies low-level circulating DNA within the exosomes. And then with that, you can really know that this patient is responding. And yeah, you can even know when there is an anemia or like a remission or recurrence or something like that. So this, I already talked about liquid biopsies in cancer diagnostics. We talked about this already. And so another thing that I talked about when I was starting is the microbiome. We see how important it is. We see that it is involved in the microbials in that. Now, like, for example, if you say HPV, it's within there. It is causing inflammation. It is causing release of free radicals, and it is causing cancer. So now studies have really gained more interest in seeing how can we use the microbiome for personalized oncology. And so since it's involved in all these things, in tumor growth, in progression, and all those things, so the microbiome is being used as a marker. So you can use it as a prognostic marker. You can use it as a marker for response to therapy. So actually now I think the research is into getting an algorithm, and this algorithm may be regional. Like, for example, the algorithm in the US may not be the one here, because you find that the microbiome in the lung or in the gut, because there is the gut-lung axis, it's not the same. So we have to also characterize our microbiome in the gut and in the lung, and then we can come up with like an algorithm, say, if you get this and this and this and this, these are predictors of disease progression. These are predictors of good prognosis. These are the predictors of resistance to treatment. So microbiome is being used for that. And also it's being used also for chemotherapy. And another thing is that also because microbiome is becoming very important and a powerful tool in personalized medicine, so it can also be used to tailor medicine to predict treatment and prognosis.
Rice? Leave when the food is ready. No, we are not going to make rice.
Mchele ugenda pagokka.
Banga, there are people who don't eat meat. Now,
Please.
David, could you kindly mute?
now there is a meat. Right?
Okay,
Yeah.
so
Everybody. And so then this sauce has to be meat and then chicken, but dry. Dry what? Dry rice. What chicken? Dry chicken.
Can mute. Okay, thank you.
all right, so since we're still talking about the newer approaches. We talked about the microbiome. Now we already talked about the exosome, but also there is more in the area of exosome in relation to cancer therapy. And because we said that these are transporters, they are cargo transporters, so they can be used and utilized in different ways. And one of the ways that can be utilized is by, one, they carry cancer cells or cancer DNA. So if you deplete tumor-derived exosomes, then you can prevent spread of metastasis and drug resistance. So you just target those that are tumor containing, and then specifically target them, and then you curtail the spread. And then also they can be used to, for example, like talked about the stroma, it has different cells in there. So if exosomes from non-cancer cells like the dendritic cells or mesenchymal cells can be also used to influence, okay, to prevent tumor immunosuppression. And also some of these can target the tumor cells to the MHC, and they can be destroyed by the immune response. Then the other is to use them as a vehicle for anti-cancer drugs, because they are cargo. So if they are loaded with the anti-cancer drugs, they can take it to that specific cell. So they can be used to target cancer cells. And because the advantage is that they are low immunogenicity, they do not cause the immune response to come out and get in disarray. They are quite calm, and so they work very well and in a precise way. So much as this research is going on and the potential is there, but there are still challenges. One, we know that, okay, not the challenge, but the advantage is that the exosomes are very safe, they are low immunogenicity, and they are precise. But there are still challenges because developing these assays to standardize the exosome and getting them out and then loading them with what you need to do, it's a lot of work. It requires a lot of money. It requires a lot of time. It is specific, and you have to have high yields. So this is a research area that is still ongoing, and more is still done to see how they can optimize the use of exosomes in cancer therapy. So exosomes are one of the nanoparticles because they are small and they can be used to target, but then there are other nanoparticles that can be used also. And these can be, there are those that are specific, they are targeted, and they just deliver the drug to the targeted cells. They seek out cancer cells and then deliver the drug to them without causing any of the healthy cells to go into any kind of disarray. And also there are those that have enhanced permeability. Because they are permeable, they can go in, and then also once they go in, they retain. So they retain the drug and they keep the drug at the site of action for a longer time and becomes more effective. And then there are those that are controlled release, where you have slow release of the drug at maybe a temperature or a pH, and this minimizes side effects. And then also they can be used to overcome drug resistance because they may bypass some of the mechanisms that cancer cells use to evade the immune response or the drugs. So these are the types of nanoparticles. There are some that are gold, there's liposomes, there's gold, there's iron oxide, and they all do different things, but most of these are still in the research phase. They're not yet out in the market. So also another emerging innovation, all the other innovations, but still there is what they call cargo-free nanoparticles. These can kill cancer cells by causing them to swell and burst with no drugs. And the beauty of this is that there will be no drugs, there will be no side effects. But also, as I was thinking about it, it's like, okay, yes, the cancer cell will burst, but what is inside the cancer cell? So once this bursts, you can have a cytokine storm where you have the immune response going into confusion because the release of so much of these things within the cancer cell, and it will cause maybe like an iris. So the challenges ahead is that much as this research is still is on and all these innovations are on, but progress is slow. Also, with the nanoparticles like gold, the iron, all those things, we don't know what the ultimate side effects would be in the long run. And also the challenge of scalability. And also for our context, affordability of these. Even when they are on the market, will we be able to afford them? So in summary, we have seen everything molecular, from oncogenesis, treatment, management of drug resistance, and the newer innovative approaches in the pipeline. Now, the question is, how far have we gone in the process of achieving precision or personalized care for our patients with lung cancer? That is a key question, and that is where our success will lie. So the question is, okay, yes, we can diagnose ably, we can diagnose lung cancer. We have all the whatever it is, we can do CT-guided biopsies, and then we do IHC, and then we do all those things, yes. But can we genotype in country? And I think I can answer that, because yes, this is very key. If we are to achieve personalized care, we have to genotype. And when you genotype, these patients come when they are late. So we need to have a system in place where the TAT, turnaround time, is acceptable. And then patients start on treatment, and then also monitor response to treatment. And also, do we have capacity for newer innovative approaches? For example, circulating tumor DNA analysis with next generation sequencing and digital PCR. As I already told you that, yes, this is a question that we can answer comfortably. We can do this in the lab that I oversee, and we have the capability to do circulating tumor DNA. We have the digital PCR machines. We have the next generation sequencing platforms. And so if we are to position ourselves for personalized care, this is available also for patient monitoring. Can we access personalized treatment based on comprehensive genotyping? So yes, you can diagnose the patient and all these things, and all the facilities are available for all the things to be done in country. But do we have the medicines to treat these lung cancer patients? That is another thing. If we are to achieve precision care, we have to have the medicines available. Then, are we able to closely monitor response to therapy? That is also key, because we've looked at all these mechanisms of resistance, and you can't just give the patient treatment and you sit back and relax. No. What if something is coming up? So there is need for serial monitoring using the non-invasive methods, and then you can ably see the patient progressing. So, however, the biggest challenge we have in our setting is that most of our patients with lung cancer do not make it through chemo. And why? It's because of late diagnosis. The patients come very late in care, and because they come very late, there's only so much you can do. And also, these patients may not afford the tests and the precision medicine. So if you have them, you have the patient, you're stuck with, can they afford the tests? Then can they afford the medicines? Then ultimately what happens, they get the one size fits all. So they get, okay, we are going to give this, this, and this. Some of these medicines are not as effective. That's why we have research to see what works best. So we see that this is now the biggest challenge we have. If we are to make better, to change the lives of our cancer patients suffering from lung cancer, we have to have mechanism of precision medicine with everything in place. That even when they report later, I've talked to some of the oncologists that some patients report really late and they're on oxygen. But if they do like there's precision medicine, and then you do the precision diagnostics and then precision medicine, then the patients will pull through sometimes. So those are the challenges we have. And I think with that, I will close my discussion or my presentation, and I think respond to some comments. And I must say I'm not the guru in this area, but yes, I've done what I could, because it's a huge topic. So I couldn't exhaust everything. Thank you.
Thank you, Dr. Immaculate. You have really helped us understand some things about genomics, mutations, And right now, I see there are some questions in the chat. I will read, I will probably check about 4 to 5 questions and then hear from you. So in the chat, there's questions about what would you say about people who get cancer treatment, improve clinically, and then on doing the PET scan, they see mets. For this presentation, are you able to comment about this, Dr. Immaculate? And there are a few other questions. And we'd also like to hear from you. If you can raise your hand and speak, that would be better.
Yeah, maybe I could respond to that first. Yeah, I think they can improve normally. We talked about the different mechanisms of resistance. It could be that, yes, this patient is taking this medicine and is improving, but then there are that intrinsic resistance because of the heterogeneity of the cancer. Then those are the mutations that will come out and then now cause the cancer to revive, to come back again. That's what I would say.
Okay, the other question in the chat is about the cost of early diagnosis if patients come late, and at what age is this late coming? Most observed in Uganda. What age do we observe the late coming?
Okay. Now the age or the time, the stage. Sorry.
I think this is more about the stage really, because age is
the stage. Well, I think most of the cancers, it goes by stage. So late diagnosis is really when there are metastases and all that stuff. So if it's early, I mean, most times actually for lung cancer, it's a little tricky because by the time someone comes with symptoms, present with symptoms, they're already like far out there. So it is very hard to get them early because it takes a while for them to manifest the symptoms. So late is late. And because by the time they come, most of them have, they actually, when they start treatment, even without the precision, most of them do not go through the chemo because maybe around 3 months, you find that maybe 50% of them are off because of the late reporting and they're already advanced and there is metastases everywhere.
sorry, yeah. So thank you, Dr. Immaculate. Let's summarize this. Maybe let me ask a question of my own. And if anyone else has a question, please raise your hand and unmute. Just one more question. But I will ask this, Dr. Immaculate. So for the clinicians on the call, you have told us about the common mutations, and with this study that we've done in lung cancer, we've established certain risk factors for patients with lung cancer. So my question is more about prevention. And in prevention, are there some, you've talked about some screening modalities such as the circulating tumor mutations. So what would you encourage us clinicians to do for patients that we suspect or we think are at a greater risk of developing lung cancer? Is there any tests that are available that you recommend in terms of turnaround time, in terms of cost, at least within Kampala? If we have patients that we suspect, there are some tests that we can do that can help us with the prevention.
Well, for prevention, I think for like any other cancers, I think screen, like, you know, how you screen and, you know, people, how they go for screening, there are those biomarker
in terms
detections.
of the mutations.
Yes. So you can like target some of those. We can do those by maybe qPCR or digital PCR, and then you just get a number. Normally, when people do that, they just do one biomarker. And if you do EGFR, and if this patient doesn't have EGFR mutation, then you're missing it. So you just have to get like a number of them and then just do a number of markers, but in a in a PCR, and then to just have a quantifying, you know, like the levels are above this, then it means that there is a predisposition for that.
All right, thank you. So the last question, Eva, I see your hand is up. Do you want to unmute and ask?
All right, thank you. I had put the question in the chat. It's about using the exosomes as a therapy, like a therapeutic in this case. So won't we worry about autoimmunity? Because I believe the link is quite high, given that we still have exosomes in the body. So how different are they?
Well, when they use these exosomes, they are specific. You know, they are those now, like when you're targeting us at an exosome that has the cancer or the tumor DNA, they are doing research so that they can be easily differentiated, because every cell in the body produces exosomes. So you have to have specific markers that will be used to tag or to identify these that carry tumor DNA, and then they can target those for depletion, because we talked about they deplete those that have the tumor DNA and leave all the other exosomes intact, because every cell has those and they come out. So it's just targeting specifically.
All right, thank you, Dr. Immaculate. I think with that, let us invite Professor Bruce Chirenga, who is our founder, to tell us more about the Lung Institute and MLI at 10 years. Professor Bruce, please, the floor is yours.
Hello, I was saying that someone could go through the slides, but about the presentation, I think this is an excellent presentation on cancer immunology, which actually should become a building block for most of our work. The interesting component is that we have our local data, which is building into this, be able to bring some of these concepts home. Because one thing, reading about these things, but also it's another to show you that actually we have been able to do this work in our setting. So thank you, Margaret, for the presentation. So about these slides, I think, I don't know whether someone is going to go through them, but we can just walk through them. That is needed to save time. What it shows here is that the Lung Institute started in 2015, and we are celebrating 10 years. And then, can we go to the next slide? Okay. This is the mission of conducting lung health research. That basically is, go back a bit. That is supported by clinical care. While we are an academic research institute, we really also think we need to provide care, and we currently have the largest chest clinic in the country, if not in the region. Okay, we can go to the next. Okay, I think this one you can skip. Next. So these are the different people who have been associated. No. I think that one where we have to show people. The other one, I didn't want to talk about it because I'm talking about myself. I don't know who was planning to present. But these are the different people who are involved in the Lung Institute. That is our board of directors. Critical about it is the representation of the Central University and the Ministry of Health and senior colleagues. I saw one of the board, the chairman of the board actually, Professor Ibinjira today attended. Maybe you will give him a minute. And then our technical committee, which involves mainly the specialists in lung science and health from different countries. Next. So this is the impact so far. We publish about 30 papers per year. And as you can see, we have seen many patients with advanced and complicated lung disease. Those are the employees that we have, and that's the grants attracted in the last 10 years. Next. So finally, we have lined up activity to celebrate the 10 years. Mainly there are three. The first one, which actually we don't list, are these series of lectures every month for the whole of this year, focusing on the 10 years impact. One of actually of the key achievements of the 10-year impact is starting this lung cancer program. Before this, actually, there was nothing about lung cancer in terms of science. Of course, treatment was happening at the Cancer Institute, but the science was really lagging behind. Then we have our symposium, the Lung Science and Health Symposium. Then we will have a dinner, which will be fundraising. And then we'll have what we call UFS, the unveiling of the foundation stone of our home here at Mulago, above Galloway. Construction has started, but we want to have a guest of honor to unveil the foundation stone. I think this is the last. So back to Treasurer, but if the chair of the board is still here, I'm only obliged to pass the mic to him, but I think maybe he has left.
Thank you so much, Prof. Yes, I actually had asked you to speak so that you could also invite the chair of the board. So I think according to protocol, please, Professor Bruce, kindly invite the chair of the board. And,
okay. So members
yes,
who are still on the call, I take this opportunity to invite Professor Ibinjira, who is the chair of the board. But also in that first picture you saw he was there, he's a founding member. Professor, over to you.
Yeah, thank you so much, Professor Chirenga, and good evening to everyone who is attending. This includes our fraternity at the college, guests, and all those who have attended from across the world, because I think this is open to everyone. So my name is Professor Ibinjira again, and I count myself privileged to have attended this webinar. I want to appreciate Immaculate for the very elaborate presentation. I'm specifically happy because we've been trying to move on a number of fronts. One is clinical care for lung health. The other one is training, and the other one is research. And I'm happy that Immaculate has proved what we are doing in research, but at the same time, her being able to present all this is a clear indication that we've done training and we are building for the next generation. I think the Professor Chirenga has talked about how the Lung Institute came about, and reaching this level, I think, is a great achievement. I know this is part of the activities that will take us to celebrating 10 years. And I think this is a clear mark that really we've done a lot in all those areas: clinical care, training, and research. The science was good. My only question I had was, Immaculate should have told us at what time these biomarkers begin to appear. Maybe you'll do it on the chart. When do they appear so that they can be targeted specifically for screening? I think we need to do a lot of screening so that we can be at the level of other developed countries, so that we are able to capture the cancers at an early stage. And I think that is one of our objectives. So I don't want to talk a lot, but I want to thank the management of the Lung Institute for planning all this and for organizing the Day of the Lung Institute throughout the year, throughout many years, and it is continuing. So I want to call upon the stakeholders of Makerere University Lung Institute, the well-wishers, and everyone who is a potential candidate of lung disease to take interest in these activities and also be around to participate on that day when we are going to celebrate the 10 years and to put a foundation stone to the home of the Lung Institute. This home will enable us to put into action many things we are not doing, like the science she was talking about, the drug development, which can be personalized or which can be used to target specific cancers and other lung diseases. All those will be possible once we have our home ready. So thank you very much. And I think I will end it at that. And I wish the Lung Institute management and the board good preparations until that very last day when we'll celebrate. Thank you very much.
Thank you, Prof. Thank you so much for those kind words. I want to also kindly add that Professor Chirenga is the founder and the director of the Makerere Lung Institute. And you have just heard from Professor Charles Winjira, who is the chair of the board of Makerere Lung Institute. I thank you both for being able to attend and for giving us your time. And I thank you, Dr. Immaculate, for the presentation. Also, as part of Makerere University Lung Institute's 10-year anniversary celebrations, we invite you to submit abstracts for the third Lung Science and Health Symposium, which will be held on the 27th of November at Makerere University Main Hall. The theme for this is a decade of advancing lung science and health for all through research, training, and clinical care. And before you leave the chat, kindly fill in the CPD link that has been provided in the chat, and so that you can be able to get your CPD points. And with those few remarks, we hope that you have enjoyed this presentation. If you would like these slides forwarded to you, please send us your email in the chat and we shall forward the slides to you. Thank you so much, Dr. Immaculate, and thank you to all the organizers for this lovely day of science. Thank
you, everyone.
you all, and thank you for attending.